complement activation. As I mentioned in the last audio clip, there are three pathways of activating complement system. One is through the classical pathway, the second is the alternative pathway, and the third is the lectin pathway. If you'll find figure 7.2 in your book, commit this completely to memory, because there's no way of learning this except to remember it. I'll go over it here. A classical pathway is one that requires an antigen-antibody reaction. So an antibody has to be produced against a specific antigen on the surface of a cell. And usually it'll take several antigen binding sites in order to stimulate this process to go forward. But we'll start with the C1 complement protein. Now C1 we'll talk about here in future clips, but it's made up of C1QR2S2. Here, I'll just go through it and then we'll try to sort it out as we go through. So once the antigen-antibody reaction has occurred at the surface of a bacterial or cell, for example, the C1 protein comes into contact with it and it activates that C1QR2S2 and causes a fragmentation to occur. So now we have that one has a catalytic activity. The C1QR2S2 with a little line above it, that means that it has some sort of enzymatic function. And it cleaves or causes the fragmentation of both the C4 and the C2 complement proteins. Now C4 breaks into the C4A and C4B. I think C4A floats off and becomes opsonin or does some other job. And in the C2, when it fragments, it becomes the C2A and C2B. So, what you have happen is when both of these fragments occur in close proximity to one another, they can form a slightly more stable complex called the C4B2A. The C4B2A becomes a C3 convertase. And the C3 convertase has enzymatic activity, and it causes the C3 protein to fragment and to form C3A and C3B. The C3A goes off and it's an opsonin or causes attraction of white blood cells or something. And then the C3B will bind to the C3 convertase to form a C5 convertase, which is much more stable. The C4B2A3B C5 convertase, let me say that again, the C5 convertase is composed of these fragments, C4B, 2A, and 3B. These form a fairly stable complex. This stable complex initiates the C5 through C9 membrane attack complex. You'll see it written in your book as MAC sometimes. And that doesn't mean macrophage. In this case, it means membrane attack complex. Basically what happens is C5 through C9 inserts itself into the membrane of an invading organism and punches a hole in it. Have you ever wondered how they put big concrete pylons in the middle of a river? That was something that I had no idea about. I'm not very mechanically inclined or engineering inclined. But I pulled over on the side of the road once and watched them do it. They literally put pylons or posts down into the riverbed and form a circle or form the shape that they're wanting to fill in, and then they pump the water out of it. When they do that, then they can put fairly dry concrete in there along rods and things and let it set up. Well, the same sort of thing occurs with the C5 through C9 complex, and we'll get into that a little bit later. The main thing to remember here, and you can rewind this over and over and over again, but the main thing to remember with the classical pathway is it requires an antigen-antibody reaction to occur. Once the C1 is activated, it causes fragmentation of both the C4 and the C2, which will form a C3 convertase. The C3 convertase is C4B2A complex. This can then cause a C3 fragmentation, and the C3b subunit of the C3 protein can bind to the C3 convertase to form a C5 convertase. So a C5 convertase is composed of C4b, 2a, 3b. I know that's confusing. I'm sorry, I don't make up the names. You just have to learn it that way. Then the membrane attack complex involves the C5 through C9 complement proteins, and they literally just punch holes in the membranes of cells. Most bacterial cells have a slightly negative charge to them. Our cells are relatively neutral with a few things associated with them, and we'll get into differences between the cell types. Now, I would like to move to the alternative pathway. Well, what if you've never come into contact with a bacteria before? Your complement system won't function if it only uses the classical pathway. Some microbial surfaces, like peptidoglycan, is a common structural feature of many bacteria. Some of these complexes can initiate the alternative pathway because the C3, which is the key to the whole complement cascade system, can react with microbial surfaces. Okay. These microbial surfaces cause it to fragment, so you have these C3a and C3b complexes floating around. Excuse me. So again, the alternative pathway is independent of antibody formation. Well, if you think about it, that makes it part of the innate immune response. The major pathway of the complement activation uses 4 different proteins. Of course, it's got the C3 protein that I've already mentioned. It's got a factor B, a factor D, and properdin. The alternative pathway is initiated by cell surface things like the foreign peptidoglycan that I mentioned. In some cases, it can be glycoproteins or lipoproteins. And when it is activated, it causes the C3 to rapidly cleave, forming C3a and the C3b. The C3 convertase is what's actually going to be causing enzymatic activity to occur. How do we proceed? Where's that page? Okay, so the factor D causes fragmentation of the factor B. The C3b and the big B, little b component of the B complex fragmentation forms the C3 convertase, which is the C3b, big B, little b, which causes further fragmentation of the C3 complex. So the C3 convertase in the alternative pathway is C3b, big B, little b. The C5 convertase is when you have C3b, big B, little b, 3b, forming the C5 convertase. And then it affects the MAC, the membrane attack complex, in the same way that the primary or classical pathway works. One of the things that really helps to control this is the fact that the half-life of C3b is very, very short. It only lasts for minuscule amounts of time unless it's bound to something else. So a lot of fragmentation has to be occurring near this cell in order for stable C3 and C5 convertases to be formed. Does that make sense? So you might have a random explosion occur here and there in the bloodstream, but they're not going to be sustained and enough of them in order to form any of the C3 or C5 convertases and do damage. That's kind of key to understanding how this is regulated, is that you have to have a number of these reactions occurring at one time. On figure 7-4, there are models of the pentameric IgM. This is kind of a planar form, but if you think about it, what's happening is when the Fab regions of at least 3 of those are going to be bound to antigen, that causes them to stick their butts up in the air, and then that can activate complement activation. But I think it takes about a minimum of 3 of those to stimulate that event. The structure of the C1 molecule is shown on figure 7-3. Look at this. It looks like to me like golf clubs with, with like a chain necklace around it. So the C1 Q molecules are the golf clubs with their little head regions. And the C1RS2, there's 2 of each of those, will wrap around kind of the neck regions of the C1Q. Then, when it comes into contact with the right thing, say an activated FC region of an antibody, then it causes the chain around the neck to kind of slip and change shape. So it causes the conformational change in the C1Q R2S2 complex, and it becomes activated, but it requires a minimum number of binding sites, which means a minimum number of FC regions. So that goes back to that classical pathway. The final pathway, the lectin pathway, is one that's really relatively new. I just noticed on figure 7-7, I didn't tell you in the alternative pathway what stabilizes that propertin that I mentioned before. Stabilizes the C3 convertase and allows it to last a little bit longer. So there, I've said it. You can read through this as well as I can. The lectin pathway usually starts somewhere with the host proteins binding to microbial surfaces. So again, it's a microbial surface, but a lectin is usually a series of sugars that form a specific shape. So it could be a glycoprotein or just a polysaccharide on the surface of specific cells. A lot of these are mannose or mannan binding lectins. Let me think. The capsular material, or the exopolysaccharide, you may hear it called extracellular polymetric substances, or slime, or biofilm. This is found in a lot of bacteria, and typically speaking, the bacteria that have the most capsule are the more virulent strains. Well, they use this capsule, and they change this capsule a lot in order to be able to hide from the immune system. But the lectin binding pathway can learn similarities, like all of the staffs have some similarities in their lectin production, or their polysaccharide production. So if you have a lectin binding region that can get on there, it can cause the pathway to move forward. And it is fairly simple to understand. You can read about it in your book, but the lectin pathway binds to these regions, and it basically uses C4 and C2, just like the classical pathway does, but it needs binding to the microbial cell wall. So the lectin has to be able to bind there. And then it activates MAST, which is a protease, which actually cleaves or causes the fragmentation of a C1-like complex. So there's another protein that looks a lot like the C1 that's found in the classical pathway, but this one only works in the lectin pathway. So from that point on, it's virtually the same as the rest of it. The C4 and C2 complexes fragment. It forms a activated C3 convertase, which is basically C4B2A. Has enzymatic activity. It causes fragmentation of the C3, and then it forms the C4B2A3B complex, which is the C5 convertase, which causes the MAC membrane attack complex to move forward and punch holes inside the membranes. There's a photomicrograph, figure 7.8, that's kind of cool, and it shows you the poly C9 complex that's formed and the holes that have basically been punched into a red blood cell. So you can activate this with red blood cells from different animals and literally use microscopes to look at the holes forming inside the cells and the leakage of the material out. The next little audio clip will be on the, let me see, 3 complement. Will be on the membrane attack complex itself, although there's not a lot to talk about there. It's basically laid out for you. And then I'll talk about regulation of the complement system, which is an important kind of concept here. You got to learn it, and then you got to learn how it's regulated. And then we'll look at some of the biological concepts and activities that occur. Before completing the chapter and talking about complement deficiencies and problems. These are some genetic diseases and problems that can affect people. Most of the time, these types of complexes are not handled by immunologists. They're more likely to be handled by human geneticists. But we'll discuss all those in future audio clips.